this channel is going to be dedicated to education primarily. Yeah, so we will be going through a lot of different textbooks. I'm pretty much just going to take notes and read, and hopefully you can learn along with me. Or, if this kind of thing puts you to sleep, then, that would be great. But yeah. So, this is the book we're going to be reading. It is a concise manual of pathogenic microbiology. So, it's actually a fairly advanced topic, but I really like this book. It is very concise, as it says, a concise manual, and it really just teaches you what you need to know on this topic. And so, as we'll learn, pathogenic microbiology. Pathogenic means it is going to attack. It is a pathogen. Microbiology just means it's a microbe or just small biology. But yeah, let's get on into it. I apologize for any preparations. I didn't get a buffer on my mic. Alright, so here we have Chapter 1, Introduction. It is generally believed that the wars are single and most destructive socio-political events with the greatest impact on the society. A glance at the estimates of casualties resulting from the major wars in the 20th century can have a chilling effect. Number of persons killed during World War I, approximately 12 million. Wow. 12 million. Number of persons in World War II, approximately 55 million. Wow. The combined total of persons killed in all of the wars of the 21st century is approximately 1 million. Wow. They weren't called the World Wars for no reason, folks. Thus, the total number of war casualties in the 20th century is estimated to be approximately 68 million. If one takes into account all war-related deaths in the world during the past 500 years, the total would most probably be less than 100 million. In contrast, during during the past century alone, more than 500 million people have died of infectious diseases, and nearly 5 billion have suffered from debilitating infectious diseases. Arguably, the number were much higher before the advent of antibiotics era and before prophylactic vaccines became available. Besides causing the social and emotional strain, infectious diseases profoundly affect economy and productivity of societies. There is no exact figure, but it is estimated that the worldwide healthcare cost during the past decade alone was several trillion dollars. Wow. Much more than the total annual budget of the United States, the world's richest nation. Yet neither nations nor societies seem to take infection is based on reports published during the past 10 years. One can very well imagine that the mortality rates were much higher in prebiotics and pre-vaccine eras. The focus of this book is on a select group of microorganisms that cause common diseases, with relatively less emphasis on the clinical aspects, though still covering the essentials. Each section deals with a group of taxonomically related organisms, or microorganisms, with an emphasis on the following. So this book is going to focus on microorganisms with an emphasis on the following. So biology of disease-causing microorganisms. Natural habitats of the causal agents. Diseases they carry, or diseases they cause, pardon me, and mode of dissemination. The laboratory. Spelled it wrong. The words were right there. That's crazy. Laboratory diagnosis and antibiotic sensitivity. Pardon that, you may occasionally hear my cat throughout the video. Okay. So moving on, we have Koch's postulate. This looks like a key term. So Koch's postulate forms the very basis of the pathogenic microbiology. The causality of almost all infectious diseases is based on the postulate, and the theories developed by Robert Koch, who is rightly called the father of pathogenic microbiology, and his contemporaries. Developed in the late 19th century, it has stood the test of time. The postulate can be summarized as follows. 1. A microbe suspected as the causal agent of a particular disease must be found in all subjects suffering from the similar disease, but must be absent in clinical specimens from healthy individuals. So, essentially, the cause of the illness, the causal agent of a specific illness has to be found in everybody that has that disease. Disease. Okay. Number 2. The suspected microorganism can be isolated from the diseased individual and grown into a pure culture. Number 3. When this isolated suspect microbe is injected into healthy and susceptible animals, some human volunteers were also reportedly used by Robert Koch. Signs and symptoms of a disease similar to the disease under investigation must develop in the infected animal. So essentially, if you have a hunch that there's a specific microorganism causing a disease, when you inject from that animal that they're suspecting, it has to be exactly the same as what was like initially taken from the person, right? So you take a bad microbe that you think is causing a disease, put it in someone or put it in an animal, they start developing symptoms. You isolate that, it needs to be exactly the same as what you put in to prove it's like the same thing, right? Okay, sorry about that. So the microbe cultured from the infected animal must be identical to original patient. Okay. Oh, so here we have some terminology. I'm not going to write all this down. This is very good to know. So, terminology. So, a side is a chemical or physical agent that kills microorganisms. You've probably heard of these terms before, but we have another list. So, we have bactericides, we have fungicides, we have bactericides, which are agents that kill bacteria. We have fungicides, agents that kill fungi. We have virucides, chemical agents that kill viruses. We have microbicides, physical or chemical agents that are lethal to a broad group of microorganisms. And then a biocide, which is substances lethal to all forms of life. Another definition here, communicable / contagious diseases. These are diseases that are easily transmitted from one person to another. So an example of this is tuberculosis. What is a disease? A disease can be defined as a state of altered homeostasis, that is, being in a state of disease. Causes and factors relating to disease may be metabolic, it may be psychiatric, environmental, infectious, immunological. Endotoxins. These are lipopolysaccharides produced by certain gram-negative bacteria, for example, Escherichia coli, so E. coli. So essentially, endotoxins are sugars made by specific bacteria that are toxic to you, and an example is E. coli. Epidemic. This excessive frequency of a disease. Etiologic agent, causal agent of a disease, which is the same as a pathogen, right? That's what causes it. Exogenous, pathogens that come from external sources outside of the body. Exotoxins, toxic proteins produced by a wide range of bacteria. Facultative pathogen, a part-time pathogen which needs to infect a host in order to complete its life cycle. Infectious disease, diseases caused by microorganisms. Okay. Mode of dissemination, medium through which causal agents of infectious disease are transmitted from person to person. Obligate pathogen, a pathogen that cannot live outside the host and is mostly dependent on a host for the ATP. Opportunist, microbes that infect a host and the host's immune system is weakened. Pathogen, a microorganism that causes a disease, which is going to be the main study in this book, the main topic. Portal of entry, the route through which a pathogen enters the human body. Reservoir, source of infection or natural habitat of the pathogen. Resident microbiota, microorganisms normally saprobe, a microbe that grew on non-living entities. This term mostly refers to non-pathogenic microorganisms. Static, a chemical agent that stops cell multiplication. They can be bacteriostatic or fungistatic. For example, let me reread that. They can be bacteriostatic or fungistatic, for example. Transient microbiota, microorganisms that are normally present in a specific anatomical site but are introduced deliberately or inadvertently. Virulence, pathogenic potential of a microorganism. That's how pathogenic, how infectious, how deadly it is. A microorganism is. Virulence factors. So these are morphological, physiological, or genetic traits of a microorganism that enables it to overcome the host's immune defenses. Examples include capsules, proteolytic enzymes, and toxins. So it's kind of a list of the terms that we'll find throughout this book. So some major categories of pathogenic microorganisms. So of course we have viruses. Viruses believed to be a bridge between the living and the non-living. Viruses have either DNA or RNA, but seldom both. Their genome is surrounded by a protein coat called a capsid. Certain viruses have an envelope, often derived from the host cell membrane during lysis and release. Multiple characteristics including type of nucleic acid, single or double strands, and presence or absence of an envelope are taken into account in the classification of viruses. Next we have bacteria. So, bacteria are prokaryotes, which means they lack nuclear membranes and other membrane-bound organelles, such as mitochondria. A shout out to the powerhouse of the cell. Anyways, such as mitochondria, Golgi apparatus, and endoplasmic reticula. Most bacteria have a cell wall, but some are devoid of a cell wall. Example, mycoplasma. Bacteria are divided into two major groups. I never know how to say this. Cocci and bacilli, or bacilli. We'll do cocci. So, cocci, which is a singular coccus, are spherical and may occur as a single coccus, as a pair called diplococcus, as in the case of Neisseria, gonorrhea. Ooh, we know that. Not we personally. Anyway, it's a common bacteria. Funny bacteria. Anyways, as a cluster, as seen in the case of Staphylococcus aureus, or a chain of several cocci, as in the Streptococcus species. The bacilli, which is singular bacillus, are rod-shaped bacteria. So this is a cocci bacteria. This is a diplococci. This is a chain. So banana-shaped vibrios, as in the case of Vibrio cholerae. Another variation in the shape is represented by the spiral bacteria called spirochetes. So, spirochetes are very weird, but yeah, we'll learn more about that. An example of a spirochete is Treponema pallidum, the causal agent of syphilis. Wow, they're really hammering home the STIs here. Certain evolved forms of bacilli tend to have rudimentary filaments, as in the case of Corneobacterium and Mycobacterium, and others have a fully developed filament with true branching, as seen in the case of Streptomyces. Yeah, these are weird. These have like little like filaments and tendrils and stuff, kind of like this. Based on their reactions to gram staining, both cocci and bacilli are further divided into two groups. So you have gram positive and gram negative. And that's depending on the color when they're stained. The gram-positive bacterial cell's wall is made of a thick layer of a peptidoglycan with some embedded digoic acid. The outer layer of the gram-negative bacterial cell wall is made of a thick layer of a lipopolysaccharide, some phospholipids, and a small amount of peptoglycan. Both groups of bacteria have aerobic, which is an oxygen-dependent, and anaerobic, oxygen-independent members. There are also several bacteria that can grow under either condition, and they are called facultative anaerobes. For a detailed discussion on bacterial taxonomy, readers are referred to Virgie's Manual of Determinative Bacteriology and numerous other authoritative sources listed in the bibliography. Yeah, that book is very dense. It's essentially an encyclopedia of bacteria. So next we have fungi. So fungi. Unlike bacteria, fungi are eukaryotes, which means they have a nuclear membrane and membrane-bound organelles. Almost all have a cell wall, which is usually made of chitin. Based on their sexual reproduction and other structural features, fungi are divided into 4 major groups called divisions or phylum. Division Zygomycota can reproduce sexually and asexually, and most, but not all, have no septum in their mycelium. Mucor and Rhizopus are examples of zygomycetes. The second division, Ascomycota, is characterized by the production of ascospores during their sexual reproduction. Ascospores are generally housed in an enclosed structure called an ascus. These are probably the most common that you'll think of, but an example of an ascomycete is a Pseudoschleria boydii. There, I attempted it. Okay. And pardon me, I misspoke. So the third division, which is Basidiomycota, is definitely going to be what you think of the most when you think of fungi. The third division, Basidiomycota, produce special cells called basidia during their sexual reproduction. The sexual spores, called basidiospores, develop on the basidia. An example of Basidiomycete is Phyllobasidella neoformans, which is the sexual stage of an important human pathogen. Common mushrooms are also members of division Basidiomycota. Fungi, which are not known to reproduce sexually, are called fungi imperfecti. It's the fourth division. They multiply vegetatively, but show considerable variation in their structure as their sexual stages are discovered. They are generally categorized as either ascomycetes Penicillium are examples of fungi and perfecti. Many fungi are also known to produce powerful toxins such as aflatoxins, aquatoxins, aminidins, and ergot alkaloids. Okay. Lastly, we have the protozoa and multicellular parasites. Protozoa. Transmission, Protozoa are unicellular eukaryotic microorganisms that belong in the kingdom Protista. Protozoa and multicellular parasites called helminths lack a cell wall. Classification of protozoa and helminths is complex, and the system is not without controversies. Therefore, authors have chosen a simple and practical approach, which is summarized in the respective chapters on unicellular parasites and multicellular parasites. Yeah, so as you kind of go up this list, it becomes more and more developed. They become more animal-like is probably the easiest way to describe it. You have viruses, which have very little genetic information, very little development. It's pretty much just a little cartridge of DNA or RNA. That's all it is. Then you get to bacteria, which are, little different types of single-cell organisms. Then you get into fungi, which are eukaryotes, and they have like cells, cell walls, and cell membranes. You have the plasma or nuclear membranes, and they have all kinds of different divisions. And then, of course, you get into protozoa, and that's when you start getting into actual little microscopic like bugs. It's kind of what you would, if you take like pond water, you see little things swimming around. That's what it would be. Protozoa, multicellular parasites. Go ahead and flip the page here. Much louder than I expected. My apologies. Okay. Okay. Transmission of infectious disease, aka the mode of dissemination. So that's important. So transmission. Aka mode of dissemination. So we have airborne. Okay. Airborne. So this is inhalation of bioaerosols. A bioaerosol contains bacteria in its center, surrounded by air and a small amount of a liquid, generally saliva. Bioaerosols may be produced due to sneezing, coughing, or talking. Depending on the force of sneezing and coughing, the bioaerosol-borne microorganism can travel up to several meters in air. Almost all respiratory tract infections are airborne. Some can also pass from person to person through the inhalation of bioaerosols. Some of the examples of airborne infections, including tuberculosis, strep throat, diphtheria, pertussis, legionellosis. I always mess up. Legionellosis disease, what I just called it. Legionellosis, influenza, and chickenpox, and a wide range of mycotic diseases such as aspergillosis, zygomycosis, cryptococcus, histoplasmosis, and coccidioidomycosis. spread by bacteria and other things. Yeah, definitely one of the most dangerous. Okay, so then we have food and or waterborne. These diseases are acquired through the ingestion of food or water contaminated with fecal bacteria or other infectious agents. The usual port of entry is mouth or gastrointestinal tract. Examples include cholera, typhoid, botulism, shigellosis, gastroenteritis, leptospirosis, hepatitis A, and a number of parasitic diseases. Zoonosis. Pardon me. The term zoonosis refers to a wide range of diseases that are transmitted via a vector, a carrier, or an infected animal. Such animals may be infected with insects, which in turn harbor the pathogen. Examples are malaria, Lyme disease, rabies, plague, Rocky Mountain spotted fever, and some of the parasitic diseases. Sexually transmitted. I hope I'm allowed to say that on YouTube. These diseases are acquired through close contact, usually intercourse. Examples include syphilis, gonorrhea, HIV, hepatitis B, herpes, and non-gonococcal urethritis. Exchange of biological fluid through oral should be included in this category. And then nosocomial infections, or nosocomial, sorry. Nosocomial infections are infections contracted during a patient's medical care, and they can include diseases contracted through fomites. Fomites are inanimate or non-living objects such as doorknobs, telephones, and computer keyboards, which facilitate dissemination of infectious diseases. Fomites contribute to many hospital-acquired infections. Examples include Clostridium difficile, associated diarrhea, urinary tract infections, and pneumonia. Nosocomial infections are, rather dangerous, especially for people who are immune compromised in hospitals. So, what are the universal precautions? At a minimum, universal precautions, that is, common sense safety measures, must be followed when dealing with potentially infectious materials or persons suffering from infectious diseases. The universal precautions require an investigator to wear protective apparel, lab coat and gloves, not mouth pipette, not talk while handling or culturing a clinical specimen, minimize socializing in the lab, not consume food or apply cosmetics in the lab. Always disinfect work area before and after work. Use proper safety cabinets for the laboratory work. With safety cabinets, you have different classes. So class 1 is for immediate preparation, the easiest. Class 2 for handling average pathogens. Class 3, handling highly pathogenic microorganisms. Class 4 is for handling deadly pathogens, mostly used at designated labs, such as the Centers for Disease Control and Prevention, CDC. I've never seen a class 4 cabinet. I imagine they're very intense. But yeah, these cabinets are just essentially different kinds of boxes and airflows that can be corded off from the rest of the world to keep everybody safe from the, dangerous bacteria they're working with or other things they're working with. So, rest of your day.